Dr. Nadir Ali. Benefits of LDL reduction are commonly overstated by drug trials.
Now, let's look at it from the standpoint of therapeutics. There is a very strong incentive to treat people to reduce their LDL levels. And we should try to find out what is the degree of benefit. So I'll start out with the most ridiculous example. You take 28,000 patients, 14,000 of them, you give them a new drug, a PCSK9 inhibitor that drops their LDL to 30 milligrams per deciliter. 30 milligrams per deciliter is very low.
Yep.
Because you and I, if you're following a ketogenic diet and we are exercising, we are fasting, our LDL levels will be 200.
Yep.
So 30 is very low. And if you treat 14,000 patients who have high blood pressure, who've either had previous heart disease or stents, who've had previous strokes, who have had blockages in their legs, this is a high-risk population. And you say, higher the risk, greater the benefit in terms of reducing LDL. And if you treat 14,000 patients and reduce their LDL to 30 milligrams per deciliter, and there is no impact on mortality. In other words, you don't reduce death rate at all. In fact, in the group that got given the medicines, the death rate is a little higher than the control group. You would say there is something wrong with this hypothesis.
Yep.
So I want to break here, and I think we have beaten LDL so much and so wrongly. And I think we have lost focus on how to improve somebody's health, because everything you do to reduce LDL is going to harm your body in terms of insulin resistance. When you remove LDL, your insulin receptor doesn't function well. Your insulin doesn't function well. Your muscles don't function well. Your brain does not function well. Although you have reduced the LDL, you have made insulin resistance and everything else worse. So why is it that we are clinging on to a hypothesis that has produced ill health in so many different ways, rather than focus on root causes, which is what you are doing? It is very gratifying to a physician treating somebody to actually look at a blood test and say, hey, I was able to drop your LDL by 50 to 70%. That's gratifying both to the patient as well as to the physician, because it is so intuitive to think that, hey, the blood vessels are clogged with fat, and this man has reduced my fat by 70%. And that's a dramatic improvement. And so you gravitate towards that. The second is the way in which clinical trials have evolved. Clinical trials have been driven by the pharmaceutical industry. The clinical trials give a very misleading magnitude of benefit. Like, for example, I will take you back to 1994 when the Scandinavian simvastatin trial was done. Simvastatin is a cholesterol-reducing drug. 1994, it was published, 4,444 patients, half on simvastatin, half not on simvastatin. This trial was completely done by Merck. Merck had the biostatisticians, they funded the trial, they collected all the data, they presented all the data. Even though the physicians presented the paper, the data was all gathered by Merck. So in other words, the degree of conflict of interest was very high. There was a significant reduction in cholesterol, I think 25% in LDL cholesterol. But the most important endpoint was that if you treated 100 patients for 1 year, you would reduce half a death, 0.6 deaths, half a death. So if your magnitude of benefit by treating 100 patients for 1 year in terms of mortality reduction is 0.6%, you should say it like that. You should not say that there was a 42% reduction in the risk of death and myocardial infarction, because that's a relative risk reduction. And relative risk reduction is very misleading. And I like to use Dr. Kendrick's example Let's say if the Texas Lotto, to win it, the jackpot is 1 in 10 million, and you come and you tell me that, hey, I'm going to double your chances. I'll improve your chances by 100%. That 100% really is from 1 in 10 million to 2 in 10 million. But if you say it as 100%, it will sound a lot more impressive than if you say, I'm going to improve it from 0.601 to 0.602. So the way in which this data is being presented to physicians is wrong, because we focus so much on relative risk reduction. And we are very happy looking at surrogate endpoints of a reduction in LDL cholesterol.
There is a very strong incentive to reduce the LDL levels. What is the degree of benefit? In a study of 28,000 participants, 14,000 were given medication that dropped their LDL to 30 milligrams per deciliter. This is very low. These 14,000 had high blood pressure, heart disease, strokes, and blockages. The higher the risk should show the higher benefit in terms of reducing LDL. However, the 14,000 participants result, no mortality benefit at all. There is something wrong with this hypothesis. We have beaten LDL so wrongly, we have lost focus on how to improve someone's health. Everything you do to reduce the LDL harms the body's insulin resistance. The methods to lower LDL cause many things not to function well, including insulin receptors, insulin, muscles, brain. Lowered LDL, but you have made everything else worse. Why not focus on root causes? It may be gratifying for the physician and patient to see the LDL number decrease. And it's intuitive to think, my blood vessels were clogged with fat, and my doctor has reduced it by 70%. Clinical trials have been driven by the pharmaceutical industry. These trials give a very misleading magnitude of the benefit. Simvastatin trial in 1994, 4,444 patients were involved. Half were given simvastatin, half were in the control group. The trial was completely done by Merck, the pharmaceutical company. Conflict of interest was very high. The results, LDL was reduced by 25%. For each 100 patients in 1 year, deaths were reduced by 0.6%. We should not use relative risk reduction of 42%. Example of misleading benefits. Suppose you join the Texas Lotto and the chances of winning are 1 in 10 million. And your friend says, I'll improve your chances of winning by 100%, which is 2 in 10 million. The way of presenting trial data to physicians is wrong. They focus on relative risk reduction and surrogate markers like LDL.